Bismillahirrahmanirrahim. Today we are going to start the cytoplasmic organelles. All the organelles which are present in the cytoplasm. Yesterday we discussed about the nucleus and discussed in detail the functions of the nucleus, the components, and individually the component with their functions. Today we are going to first of all describe what are the components, what are the different organelles present in case of cytoplasm. First of all, the cytoplasm is composed of cytosol and the organelle part. The cytosol is the aqueous part of the cytoplasm, while other organelles like ribosomes, different types of ribosomes, Golgi apparatus, lysosomes are present in the cytoplasm. So starting from the ribosomes are large molecular weight structure found in the cytoplasm. Means ribosomes are not very small organelles, but they are the large molecular weight structures. As the names of the ribosomes indicates, these are the structures which are responsible for the synthesis of different types of proteins. So these ribosomes are mainly involved in the process of translation. So these are micro machines for protein synthesis. Protein synthesis means translation. Translation is the process where the different types of RNA are combined together to form the protein. These are also present in mitochondria, rough endoplasmic reticulum, and free ribosomes. So this is the structure of the ribosomes. Here you can see the ribosomes are the protein factories. Different protein factories in connection with nucleus are present, and some of them are associated with rough endoplasmic reticulum. Means the structure which is extended from the ribosome is called as the rough endoplasmic reticulum, and these free ribosomes are the cells proteins factories. Cells protein factories means these are composed of two subunits, the smaller subunit and the larger subunit. You already have a little idea about these smaller and larger subunits. These ribosomal subunits are different in prokaryotic and eukaryotic cells. In case of prokaryotes, they are of different sizes, and in case of prokaryotes, they are of different sizes, and their sizes are based on the basis of their sedimentation coefficient. It means when these ribosomes are separated, the weight is based on their separation. So these two subunits are smaller and larger subunits, and during the protein synthesis mechanisms, these both unite together to form the release of a newly synthesized protein. Means no smaller subunit is responsible for the synthesis of protein, while the other is also not responsible for the synthesis of protein. When both are joined together, they both then responsible for the synthesis of proteins. Smaller subunit is 40 Swedberg unit, while the larger subunit is 60 Swedberg unit long. Smaller subunit is responsible for the attachment and translation of messenger RNA, while the larger subunit is responsible for the release of newly synthesized proteins. Here you can see the larger and the smaller subunits. These larger and smaller subunits, when joined together, then they are going to make the protein. Here you can see the structure of the protein. In this structure, the thread-like structure of protein is protruding from these Svedberg units, and when these both ribosomal subunits join together, they are associated with the synthesis of thread-like structure of proteins, primary structure of proteins, which is made up of linear sequences of amino acids, and then these amino acids, when they combine with the peptide linkages, they lead to the formation of a protein, the primary structure of protein. So the diameter of the ribosome is about 10 nanometer. The upper one is the larger subunit, the lower part is the smaller subunit, and this messenger RNA is going to be made, which carries the message from the nucleus, and then it is transported out of the nucleus to the ribosome, and then the protein synthesis is going to be taken place in the cytoplasm with the help of these ribosomal subunits. Then coming towards another structure which is known as endoplasmic reticulum. It is continuous system of interconnected membrane bound channels. Channels means you can relate it with the channels which are going to be out from the rivers. These are the same as the channels protruding out from the ribosomes, and they ramified throughout the cytoplasm. There are two types of endoplasmic reticulum: rough endoplasmic reticulum and the smooth endoplasmic reticulum. Based on the name of their different types, their functions are different. The endoplasmic reticulum is seen in this way, and some which are without ribosomes are called smooth endoplasmic reticulum, and some which are attached with the ribosomes are called as the rough endoplasmic reticulum. The walls of the endoplasmic reticulum are called as the cisternae, while the space inside the cisternae is called as the cisternal space or the lumen. So here you can see the endoplasmic reticulum is extended like the channels throughout the cytoplasm. The nucleus part is separate, while the other part, which is cytoplasm, is composed and is made up of the endoplasmic reticulum mainly, and the endoplasmic reticulum is spreaded like the channels, which contains different components like cisternae, the lumen, which is also known as cisternal space, and the different classes, which could be rough endoplasmic reticulum and the smooth endoplasmic reticulum. So here the function of ribosome is not ended. The ribosome continues performing its function. The protein which is going to be synthesized on the ribosomes are being synthesized on the rough endoplasmic reticulum also, and then these proteins are going to be transported to the Golgi apparatus. We will see it in later. First we will discuss some of the functions of endoplasmic reticulum. So endoplasmic reticulum is the main site for the protein synthesis. Which endoplasmic reticulum? The rough endoplasmic reticulum, because the ribosomes are only present on the rough endoplasmic reticulum, and proteins are being formed here, moved to the other organelles. There are some other types of proteins called chaperones are also present in this endoplasmic reticulum. What are these chaperones proteins? These are heat shock proteins. If some of the proteins are going to be denatured by some of the effects of the environment, then these heat shock proteins protect them from being degraded. These are heat shock proteins. These can neglect the effect of the shock and protect the protein. Some of the carbohydrates are also synthesized in the protein glycosylation. These protein glycosylations are the signals which are going to be attached with the proteins, where they are going to be function later on. So first of all, in rough endoplasmic reticulum, the proteins are synthesized, they are protected, and they are being labeled with different signals to be directed to different tissues for their functioning. Some of the functions of smooth endoplasmic reticulum is the lipids, cholesterol, and steroid hormones are going to be synthesized in this smooth endoplasm, because these are not protein in nature and we don't need any kind of ribosomes here. So these other than protein biomolecules are going to be synthesized, like lipids, cholesterols, and steroid hormones. And some of the carbohydrates and proteins are also synthesized in the smooth endoplasmic reticulum. Some of the phospholipids are synthesized, and one of the main functions which is frequently asked in the MCQ is the metabolism of xenobiotics. Here in the smooth endoplasmic reticulum, we have a system known as cytochrome P450 monooxygenase system. This cytochrome P450 monooxygenase system is used or is made to metabolize the xenobiotics. What are these xenobiotics? These could be different drugs. These could be any foreign compounds, like drugs, like pollutants, like other chemicals which are present in different foodstuffs, etc. So when we eat these xenobiotics, when we engulf, when we intake these xenobiotics, these xenobiotics are going to be neutralized with the help of cytochrome P450 monooxygenase systems. Neutralization means this system made xenobiotics polar or excretable, so that the effect of these foreign compounds could not be harmful to the body and they could be easily excreted out of the body. So overall classification of the function of smooth endoplasmic reticulum includes the synthesis of lipids, the synthesis of carbohydrates, some of the proteins, some of the phospholipids, and the main is the metabolization of xenobiotics. So what is the xenobiotics? Xenobiotics are the foreign compounds. Foreign compounds could be dangerous. So smooth endoplasmic reticulum has the system. The name of the system is cytochrome P450 monooxygenase system. What this system do? This system neutralizes the effect of xenobiotics. So going towards Golgi complex. बचपन से सुनते आ रहे हो, Golgi complex is for the packaging of the protein, is for the packaging of protein. जो भी प्रोटीन आपकी सिंथेसाइज हुई राइबोसोम्स की रेफ एंडोप्लाज्मिक रेटिकुलम की मदद से, it is going to be transported to the Golgi complex. What is this Golgi complex? It is a flattened membranous cisterns or the sacs arranged in a stacked form with associated vacuoles and vesicles. So this is the proper definition of Golgi complex. You have to memorize it. It is made up of sacs which are arranged in a stacked and associated with vacuoles and vesicles. Here you can see the Golgi apparatus. The central part is known as pneumal, while other vesicles are also present which are associated vacuoles or vesicles. And this all structure is known as flattened membranous cisterns or the sacs. This is the definition. Going towards the function of the Golgi apparatus, it is used for the processing and packaging of proteins which are synthesized in endoplasmic reticulum. I told you before, all the proteins are going to be mainly synthesized in the rough endoplasmic reticulum, and then they are going to be processed and towards the Golgi complex for their packaging. So the site for the protein synthesis is going to be attached with different carbohydrate side chains like glycoproteins, glycosaminoglycans, and Golgi apparatus is also a site for the production of primary lysosomes. What are these primary lysosomes? We will discuss in detail what is the difference between the primary and the secondary lysosomes. Lysosomes are the main organelle which are going to be used for the digestion of different biomolecules. So primary lysosomes are different from secondary lysosome. We will discuss in detail in the topic of lysosomes. Here we are going to end the lecture by studying 3 types of organelles. I'm going to brief all the organelles we studied today. Cytoplasmic organelles like ribosomes, endoplasmic reticulum, and Golgi apparatus. From tomorrow we will start discussing it from the lysosome and onwards. What you people do, you have to memorize. You have to memorize the structure, the functions, and the associated functions of the organelles. The proper definition, the structure, and the functions associated with these organelles. So here we are going to end the lecture. Wish you best of luck.